Hi everyone, welcome to my poster presentation. My name is Lauren Kubis. Today I'd like to share some results with you on a project studying children with autism spectrum disorder. So we were interested in investigating relationships among brain network dynamics during resting state, task states, and behavioral measures of flexibility in children with autism compared with neurotypical children. So we wanted to investigate this because little is known regarding the relationship between dynamic brain function and flexible cognition in children with autism. So to do this, we had 27 neurotypical children and 17 children with autism first complete a 10-minute resting state fMRI scan, and then while in the scanner, they completed 4 runs of a task. And I'll go into a little bit more detail on the task right now. So the task was an attention switching paradigm that tested their ability to flexibly shift their attention between stimulus dimensions. The dimensions consisted of either color or shape. In each trial, one of the 3 objects differed from the other 2 in either shape or color. They're presented with shape blocks, color blocks, and also mixed blocks. And the mixed blocks consisted of either shape or color trials. The participant was asked to select the unique shape or color in each trial with a button press. We were interested in how the brain network dynamics changed in each task run and how it related to their behavioral performance on the task. So in order to do this, we first had to pre-process the fMRI data, and then we used ICA to hand denoise the data, and finally we used post-processing procedures that are seen here. We wanted to then investigate 6 specific regions of interest that were selected due to our interest in investigating brain networks previously shown to observe flexible behavior and behavioral inflexibility in autism. Specifically, we chose regions of interest of the lateral frontal-parietal network, also known as the central executive network, the medial frontal-parietal network, or the default mode network, and the mid-cingulate insular network, also known as the Salience network. We extracted the time series from these regions and all of our participants to use in our dynamic analysis. To complete the brain network dynamic analysis, we looked at the co-activation patterns among those 3 networks. This method also allowed us to see how many times the brain switched between the different co-activation patterns, how frequently the co-activation patterns appeared, and how long a co-activation pattern was instantiated for, or its dwell time. After inputting the time series of the 3 networks during the rest and 4 task runs into one matrix, we used k-means clustering to obtain the co-activation patterns from the rest and 4 task runs, which resulted in these 5 co-activation patterns seen here. Each co-activation pattern, or CAP, consisted of different combinations of network nodes. For example, CAP1 had high co-activation among the nodes of the lateral frontal-parietal network, and so on. Our first step was to see if there were any brain and behavior associations between the brain dynamics and behavioral measures. After a series of Pearson correlations, we found that dwell time of the medial frontal-parietal network was associated with social abilities in both neurotypical children and children with autism. As you can see, a higher dwell time was associated with a higher score or better social skills on the SRS-2, which is a parent-report measure of social abilities in children. Overall, the dwell time of the MFPN was associated with social abilities. We then ran a series of mixed model ANOVAs to test for interactions between children with autism and neurotypical children. For frequency of occurrence and dwell time of each of the 5 co-activation patterns, the models consisted of a 2 diagnosis by the 5 conditions, which were the resting state and the 4 task runs. For the transitions, each of the 5 co-activation patterns, we ran a t-test for the resting state transitions between diagnostic groups, and then a 2x4 mixed model ANOVA for the transitions during the 4 test runs. We first saw a quadratic interaction for the dwell time of CAP3, in which the MCIN was co-activated with the LFPN. We found children with autism initially spent more time in CAP3 compared to neurotypical children for task runs 1 through 3, then spent less time in the CAP3 during task run 4. We next saw a linear interaction for the frequency of occurrence of CAP1, where the LFPN was co-activated with itself. we found children with autism initially had less occurrences of CAP1 compared with neurotypical children during the first task runs, then had more occurrences of CAP1 in the last task runs. We also saw in both instances, children with autism had significantly different brain dynamics compared with neurotypical children during task run 4. What was interesting was that although children with autism showed significantly different brain network dynamic patterns, primarily in task run 4, they performed similarly on the tasks as the neurotypical children. Both children with autism and neurotypical children had over 80% accuracy on the task trials throughout all the task runs. This suggests that children with autism may neurally compensate to reach optimal behavior. So just to summarize, we found brain dynamics of the MFPN may index individual differences in social abilities, and children with autism may neurally compensate to obtain optimal behavior. Thank you so much for visiting my poster presentation, and I hope to chat with you more, so feel free to message me or we can Zoom also. Thank you so much. Bye-bye.